Hey guys, this is Dr. Cummings from Point Loma Nazarene University again, and we're in the middle of our series on reading and making sense of primary literature articles in science, and in particular microbiology, which is my area of expertise. We've been using this paper from Botts et al., Frontiers in Microbiology, as our example. What I want to do in this video is talk to you about the importance and the technique of diagramming workflows when you're looking at the methods section of a paper. Now, if you go into the paper really wanting to understand the nitty-gritty details of how they did this particular study from start to finish, one of the best things you can do is read through the materials and methods. And I would say start by reading through them, in this case, broadly at a high level, where you're not trying to keep track of, like in this study, you're not trying to keep track of how many micrograms per mil of tetracycline was in there, but you want to know that they added tetracycline, right? So you're going to look at these things at a broad level first, and then begin drawing essentially a flow chart of the steps involved, not every single detail. So this is different from a workflow, is different from a protocol. In a workflow, we're trying to really map out in our minds and visualize the work that they did and the order in which it was done. That kind of a workflow can often be all you need to be able to jump into the results section. The other benefit to this kind of workflow mapping is, is when you're doing your own research. Right? You're in the laboratory and you're moving forward with a research plan. Don't ever do that without a workflow, right? I tell my researchers that they should spend more time planning out and workflowing and strategizing and preparing for experiments than actually carrying out their experiments. Because if you do that, you're far less likely to have to repeat anything. You're not going to finish an experiment and then look back and say, oh, I really needed to have done this first, so that I would know what parameters to use for this next one. And so instead of guessing, I should have worked out the experiment over here first. So we spend a lot of time at the marker board, and we'll put up an entire workflow sometimes for the next several months, and we'll have a lot of our conversations standing in front of that workflow, adjusting and modifying that workflow. It's the map. It's the map, but it's a flexible map, because if we run into a problem or we make a realization that, hey, I got to change something, I got to do this first, or going into this step isn't right, let's go ahead and use a different protocol instead of that particular protocol, that workflow can really help. So let's skim through the materials and methods section of this paper, and then I'm going to show you, I'll actually workflow the first half of it for you to give you an example of how you can use this to really visualize and understand the experiments that were done in a paper, as well as for your own research, how to begin putting your vision on an iPad or a marker board or wherever it happens to be. So we're in the materials and methods section. First thing we've got is description of a study site. If you remember from this paper, they captured 4 multi-drug resistance plasmids from sediments of an urban coastal wetland and then characterized them. So here's a description of the study site. Skimming. They're just describing it. And it's important to get to that stuff eventually, but I want to see what they did. Oh, they captured surface sediments. I don't need to know it's 50 grams from the top, sterile spatulas at this point for workflowing. I don't need to know that. I just need to know they grabbed surface sediments. Okay, so that first part is surface sediments. Antibiotic abbreviations, I'll refer to that when I need to. And then I can see they've got 2 plasmid capture methods, an endogenous method and an exogenous method. I should probably skim through those and get a sense of what was going on for each of those. So in the endogenous method, looks like they took sediments, added it to LB broth, added antibiotics to the broth, and then cultivated the bacteria, and then streaked a colony on EMB agar that also had antibiotics multiple times till they had a pure culture. Then they extracted plasmids from it and did a heat shock transformation into E. coli. So that's their endogenous method of capturing a plasmid. Then their exogenous method, looks like they took the sediments, washed the bacteria off the sediments gently to keep them alive, took that bacterial suspension and then actually mated it with an E. coli. Mating, so here's, sometimes you're going to come up with language in this that doesn't make sense. Remember, the scientific journal articles are scientists talking to scientists. And so there's a lot of assumptions about the language. You may need to go dig up a lot of terms to build your vocabulary here. So they mated, meaning they encouraged conjugation to take place between these bacteria that they gently washed off the sediments and two lab strains, an E. coli strain and a Pseudomonas strain. In other words, they had donor bacteria and they were trying to encourage conjugation into these lab recipient bacteria. Let's keep looking. And then it looks like they did a confirmation step to make absolutely sure that what they had was trans conjugates, recipients of their plasmid. And they did that with rapid PCR and then plasmid prep with gel electrophoresis. Now, once they had all of these plasmids captured, then they went into sort of the second half of their study, which was testing for antimicrobial susceptibility, determining plasmid copy number, and then this plasmid genome sequencing and annotation analysis. That's going to require a whole separate workflow of its own because it's long and it's complex. There's a lot of different methods they used, a lot of software they used, a lot of things they were trying to accomplish there. Let's go ahead and map just the first half of this. Let's create a workflow for how they captured their plasmids in the first place. So I'm going to jump over to a little video that I made here of me creating a workflow. I just put the paper in front of me and I had my iPad out and I opened up some software and started writing out a workflow for the first half. So the first thing they did was they collected their sediments, and that's where this whole thing is going to start. Put a fancy box around it, and then from the sediment they went in two different directions to capture their plasmids. They had an endogenous route and an exogenous route. So let's look at the endogenous route first. First, they took the sediments and they added it to LB broth with particular antibiotics that they were interested in identifying resistances against. Once they had growth from that, they started doing streak plates on EMB agar with the same antibiotics to keep the selective pressure on. Once they had colonies there, they did a plasmid prep on the colonies. And then they heat shocked the plasmid that they were able to pull out of those EMB agar colonies. They heat shocked it directly into a laboratory E. coli strain. So now the plasmid is isolated from the mix that was in the sediment. And to confirm it, they did a plasmid prep on their E. coli along with gel electrophoresis to visualize that they had captured indeed a plasmid. This was the confirmation step. So now they've got plasmid by the endogenous method. At the same time, in parallel, they were working on an exogenous capture method, where in this case, they took the sediment bacteria and instead of enriching them in LB, they removed them gently using sodium pyrophosphate, I think it was. That's not important yet. They took that suspension of bacteria and they mated them with a lab E. coli, or there was also a Pseudomonas putida in there. Remember, mating means they encouraged them to conjugate. And they should have gotten what are called transconjugants, and they have to confirm those. So they used something called rapid PCR. We're going to have to look that one up. It's a fingerprinting technique. And at the same time, they also did a plasmid prep and gel electrophoresis to visualize the new plasmid in their E. coli strain. You see how that works? Now, if we wanted to continue, we'd have to have branches coming off. We'd have to say, okay, now that we've got these plasmids purified and in laboratory strains separated from the big mix that was here in the sediment to begin with. They're now individually separated in cultures of bacteria they can work with. Now what did they do? Now they've got these other steps, the characterization of antimicrobial susceptibility testing and determining plasmid copy number. And then there'd be a whole separate workflow for the sequencing, the genome sequencing of the plasmids themselves, and all of the annotation, right? Mapping out, again, you got to look up terms, mapping out all of the genes that are on there and any analysis that they did. That's going to take a whole separate flowchart. But flowcharts, these workflow charts like this are so valuable to get a big picture view of what's going on in the methods. In the next video, we're going to talk about, okay, how can we take one of these boxes, one of these steps in the flowchart, and map it out as a protocol? How can we diagram it as a step-by-step protocol that we could actually take into the laboratory and sit down and carry out that experiment? With this workflow, you can't sit down and carry out the experiments just yet. This is big picture mapping. But if we want to do one of these things, like we want to do this mating experiment here, we've got to look at it precisely. What do we do and in what order, what volumes, what concentrations, how long, what temperatures, all of those little details. That's going to be in the next video.